thank ABTA for putting on these seminars, and thank you everyone for joining. Really what I want to talk about, even though it says novel designs, I think the goal about everything we do is to come up with therapeutics. And the path that we come up with therapeutics and allowing them to be used by others is a process of clinical development. And in the end, we're looking to try to get approval from the FDA to be able to use in patients because we showed that there was a meaningful benefit. And I want to go a little bit through the process of how that works and where we've struggled in the past, and maybe different things that we can do to help us move forward in a more efficient way with developing drugs for glioma and glioblastoma. And the reason we have to develop drugs is because we don't have early detection. This is just an example of a tumor that was identified after the patient had a surgery, and the patient then 3 months later has a very dramatic increase in size of the tumor. These are the types of things that occur, and we've even had patients where there was no change on the scan that led to these types of effects. So getting MRI scans regularly doesn't help us detect it that much earlier. And when we find it, it's advanced, and there's no known cures, no known causes, so we can't prevent it. So those two things, lack of early detection and not being able to prevent it, are probably the two most important things. And so that means once we see it, we have to know how to treat it. And currently, the therapies that we have are not curable: surgery, radiation therapy, and chemotherapy. And even though we know a lot about these tumors, we know, based on the cancer genome evaluations that have been done, that there are a lot of specific molecular abnormalities that exist in these tumors. But the only ones so far to date that we know that are either prognostic or predictive are IDH, which is isocitrate dehydrogenase, which usually portends for a better outcome in glioblastoma and is usually associated with lower grade tumors, or MGMT, which is methylguanine methyltransferase, which is a predictive biomarker for the use of temozolomide, and that represents about 35% of glioblastoma. So the different therapies that we would consider using, we kind of batched them into this group of, are they cell autonomous or are they involving the microenvironment as their primary effect? And the cell autonomous ones means that it affects the tumor cell itself. Those could be like DNA damaging agents like chemotherapy and radiation therapy, or pathway inhibitors if there's an activated receptor sending signals down that we can inhibit it. Or new metabolic inhibitors, things that are affecting kind of the main metabolic and biosubstrates that are needed by the tumor, such as glucose and making lipids and proteins, or energy molecules like ATP. Or these death enhancers, which are things that cause the cell to die. And even though chemotherapies may do that, there are other agents that may push the cells along the path to cell death. And in the microenvironment, there are two main areas, immunotherapy, which the immune system, of course, is involving the microenvironment around the cells, and if they can be activated to kill the tumor, that's great. But there's also a microenvironment of immune suppression that occurs, all the macrophages and cells that exist around the tumor that prevent the immune system from being effective. And then there are processes like anti-angiogenic approaches to try to prevent blood vessels from forming, because blood vessels are necessary if the tumor is going to get large or get larger over some period of time. There requires new blood vessels to bring nutrients in. So those are the different types of therapeutic options, and they all have to go through this path of development, of clinical development, where there's a process where we either are deciding to use it because we found a unique target on these brain tumors, and then that we develop a drug to that target, and we go about with medicinal chemistry creating that we try them out in models like cell lines or lines that are put into animals to try and better understand whether there's an impact from those therapies. And then they begin to kind of bring up enough drug and have it be approved by the FDA for human use, not approved for use for glioblastoma alone, but just approved for human use. So then it goes through a process of phase 1 evaluation where we do dose escalation and ask the question, is the drug safe? Then we want to have an understanding as it moves through, and we find out that it's safe, and we know what drug level, what drug dose we can use. Does it provide something about value to the patient? Do we see, is it shrinking tumor? Is it prolonging the time that it takes the tumor to grow? Is there something that we're seeing that is providing value? And then finally, the phase 3 setting is, does the drug, does it have some value to show that it is better than the standard of care? And this is the process that most cancer therapies go through. And the part of the problem is there are a lot of other questions that have to be asked in brain cancer that don't necessarily have to be asked in other tumor types. And that includes this area that drops down here, which is where we do a little bit harder work once we know the drug is safe and we know the dose that we're interested in. That we want to begin to look at, does it get into the brain? Does it hit the target we're interested in? How should we be dosing it so it maximally has an effect on the tumor? So these are the types of things that we would address in this kind of separate setting that we kind of either call a proof of concept or an early phase evaluation. So it also costs a lot of money, which it may seem a little silly to go through this, but the reality is the way that drugs go through here is that they get funding from either venture capital or they raise money to be able to run such studies. And it's thought that it takes about almost a little more than $1 billion to kind of take it through this entire path. That this early part, after they look at a lot of different drugs, a lot of different possible drugs that could be used, and narrow it down to 1 or 2, and that show that they're efficacious, cost a significant amount of money. To do these early phase to find out if the drug is safe, costs a couple $100 million, and then to go through this longer phase 2, phase 3, requiring lots of patients, costs a lot of money too. That gets up to in the range of $300 to $500 million. So that's a lot of money. And it's a lot of money if the track record for glioblastoma isn't so great. So when people have tried therapies and we haven't seen the benefit, it becomes harder for people to think about making a bet like that on glioblastoma. So this is something that has to be changed in some way. The financial, the cost that one has to incur to go through this has to be adjusted in some way. And I hope to show you some novel ways of considering that we're going to move forward with to try to improve that. So as I said, there's a lot of drugs that have been tried in glioblastoma, and we've had a lot of things that haven't worked. And we should be thinking about why haven't they worked. And there's things having to do with the drug itself and the disease itself. And then there are things having to do with how we go about trying to decide whether these drugs or therapies are effective or not. So we have this issue about the blood-brain barrier, that it's there to prevent toxins from getting into the brain because the brain needs to function well at all times. We know that the blood-tumor barrier is disrupted. There's many times contrast enhancement, but still there's a limited amount of drug that gets through. We know that as these tumors grow, they kind of take on their own ecosystem, and in different areas of the tumor, they could look slightly different. So there's this molecular heterogeneity that exists. I won't get into this copy number issue about that this disease isn't always about mutations, but lots of copy numbers of genes or the absence of them. But one of the other things that limits our ability to be effective with our therapies is that the brain is so important to us. It's critical for our function, and we can't surgically remove it. We can't simply radiate it because we need it to function. So those things interfere with our ability to control the tumor. And then we have this issue around that the brain doesn't like inflammation in it. And it's created an environment for it to have limited inflammation and to be an immune suppressive environment. The other thing that's important for if one is going to have an immunologically active tumor is that tumor needs to look different than the rest of the body. And when there's a lot of mutations, it looks different because there's unusual proteins coming out of it. But when there are very limited mutations, that becomes a little bit more of a problem. And the brain is not a great place for us to have antigen presentation. We don't have a lot of lymphatics. And again, just even the tumors growing in the brain create a systemic immune suppression that occurs. So there's a lot of problems that occur with immune therapies. And in reality, as I said before, it's hard for companies to come up and say, I'm going to develop this just for brain cancer. That means that many of the drugs that we get have been developed for other cancers, have been shown to be effective, and now they want to be tried out in brain cancer because it isn't quite as expensive to go from zero to approval with a brand new drug. But if you have a drug that has been approved for other causes like lung cancer, breast cancer, there's an opportunity to use those drugs in brain cancer. But the downside of doing that is that it doesn't always make sense for brain cancer. For instance, we have EGFR mutations that exist in brain cancer. They also exist in lung cancer. But those mutations are completely different in the location of where they occur in lung cancer than they do in brain cancer. So the drugs that fit nicely into the mutations for lung cancer really don't end up fitting that well for brain cancer. So these repurposed drugs don't always work so well. I think it's important to kind of start off when we think about what is it that we're looking for, what is the FDA really looking for in a drug for it to be approved and to define treatment benefit. And that is that it has the impact of the treatment on how a patient survives, feels, or functions in daily life. And that is how we measure the benefit, is through these three features. Again, this is the same thing as clinical benefit. These things could be measured directly, such as looking at the tumor or looking at the patient, how long are they living for, or be how well is the patient functioning, asking the patient or asking a family member how well the patient is functioning. And this information, it provides the labeling for the promotion of the drug so that it could be used. So it says, this drug prolongs survival. So in general, particularly in glioblastoma, although everybody would want to have the opportunity to feel better or function better, we basically need patients to live longer, and that needs to be the primary endpoint that we need to go forward with, where we need to improve our ability to control these tumors. So there are actually these other surrogates that exist for survival. And although these surrogates down here, which is response rate, did the tumor shrink, or progression-free survival, how long did it take for the tumor to grow, these are not normally what is used for FDA approval, but they could be a surrogate for survival, which may allow for an accelerated approval process. So all of these things are kind of what we're looking for. But in the end, our goal is to prolong survival. And then if we do that, we'll work on feel, function, and the other aspects that are important. This is just a sampling of drugs that we have evaluated, and all of them ended up not showing a benefit, and all of them had some early data that made people think this was a good therapy. We're going to do a phase 3 study and spend that hundreds of millions of dollars because these are the data that look good. And the reality is that's what this rationale is, and almost in every single one of these settings, the problem is that they did a study where they just included one arm. That means they gave the drug, they saw how patients did, and they said, this looks better than what we normally would have thought, so this must be good. And that was the decision for going forward. And I want to get to some of the issues around why that's a problem. Why it's a problem if we just give patients the one drug and see how things go. And so what we saw there is that we gave patients just the one drug, we saw what they did, and it ended up being a false positive, meaning the signal that we saw was false because it didn't show up in the phase 3 study. And so one of the biggest problems that we have is the poor use of historical control. and the opportunity that that single-arm trial overestimated the effect so that we had some control. We said, this is what we normally expect. We gave the drug and they did better, and we said, oh, that must be great. But we didn't do a randomized study to know what the control really looks like. We could use an early endpoint in exploratory setting that really doesn't correlate with the eventual endpoint, such as overall survival. And I will get into an example of that with bevacizumab, where response rate and progression-free survival in the end didn't give us the insight into what was happening with overall survival. And again, part of the problem that people have is they're not sure that they really want to fix this. They want to have opportunities to do phase 3 studies. And even if it's a false signal, why not go through with it? Let's see what happens. Well, the other side of this is these single-arm studies may miss signals. They may miss the opportunity for a drug to be effective. So if we used a historical control and had a better outcome than we thought was going to occur, and that single arm in the end we looked at the data and it was an underestimate of effect, then that would be, we would lose the opportunity to evaluate that drug. We also lose the opportunity if we have a subclinical effect, and that is that maybe we didn't have better survival, but the dose we were using wasn't high enough for us to have that effect, or we should be using combinations. But going through this evaluation takes more time, and the more time we take affects how much money is spent. So the other opportunity with these false negatives is that maybe what happened there is there was a small population of patients with a specific subgroup that exists that has a unique molecular marker that it could account for what was for the benefit of this drug, even though it wasn't seen because they didn't go and look at kind of the details of the molecular marker on all those patients. So I want to give you an example of how we could get into trouble when we use single-arm studies. And this is an example of a vaccine study. And usually what vaccines do is we have the patient here, they have their surgery. The vaccine may be used from that tumor sample. But then the patients then go on to receive radiation and chemotherapy, and it's uncommon that the vaccines are given during that time. They're usually given after radiation and chemotherapy. And then they start, they randomize the patient, they start the treatment. But if we're comparing the vaccines that have all these patients going forward compared to all of these historical patients, we actually have patients who drop out even before they can get here. So just that process makes the treatment arm of the vaccine look better than the control arm. That could become a problem. Now, if we have more selection in this vaccine group, like they had to have a complete resection of the tumor because a vaccine can't deal with residual tumor, we now can say, well, in this control group, we now will be looking and taking only those that had gross total resection, but we still won't have a benefit because we have this survival lead time in here that interferes with it. Finally, we could say, okay, we're going to start everybody at the time of the of the vaccine. So now things are looking a little bit better. But finally, even we have to consider, well, there could be no tumor after radiation and chemotherapy and no steroids. And that all of a sudden changes the population. But if you make your control like that, it will start to look better. But the cleanest way of doing this is to have a true control. And we'll get into the issue around that at how that may seem like a burden to some patients to have to go on a control to get this information. But otherwise, we make bad decisions. And the bad decisions lead us down this path where we make, and companies make these big financial commitments that end up being negative and draw and push companies away from wanting to develop drugs in brain cancer. So we have to be aware of these single-arm studies that they may not be providing the benefit that we thought we would see. And I think that, you know, those of you who have been around through a lot of different trials in the past know that patients who enroll in these trials and find early benefit, that they could be hyped out to a significant degree and yet not show to be beneficial in a phase 3 study. So this is the example of where bevacizumab was problematic. Well, bevacizumab as an anti-VEGF therapy, it grabs on to this vascular endothelial growth factor that is the cause for leaky blood vessels, And leaky blood vessels is what causes the contrast to go through the tumor. And if we block those leaky blood vessels, we see a remarkable response. And I don't think anybody would say that there isn't a value of taking somebody's brain that looks like this and giving something like bevacizumab and have it look like this. But we saw response rates like we never saw before. We saw progression-free survival like we never saw before. And yet in the end, there was no difference in overall survival. So all of these studies are progression-free survival. This is the percentage that are progression-free over time. And you could see a placebo control arm versus the treatment arm, placebo arm versus the treatment arm, placebo arm versus the treatment. Bevacizumab always did better with regard to progression-free survival because it made the scan look better. But now when all the studies are done and they look at survival, they see there's no difference in survival in any of these. And you would think that this should have been a good bet because we were seeing these kinds of responses. But in the end, we didn't see them, and it became problematic. Now, in the end, this is still a beneficial therapy, but in terms of it having an impact on overall survival, I think that's unfortunately didn't occur. Another thing that can happen is that when we're trying to do these evaluations in patients, we can get, again, false signals that occur. And one of the false signals is determining that progression occurred when it didn't really occur. And this is a problem that occurs with investigators making decisions. So here's a patient that has a surgery. This is the post-operative scan. They undergo radiation and chemotherapy, but they decided they didn't want to get a scan until the second post-surgical cycle of temozolomide. And they had this. And when they compared it here, they said, oh, that's definitely bigger. But what they didn't say is what happened during this time when there was no treatment, and then even during treatment, could there be inflammation that occurred? And they called this patient progression, but then they waited, gave another cycle, repeated a scan, and said, yep, that's still progression. But when you compare this scan to this one, you see that it's starting to shrink on its own. They went on an experimental therapy. It continued to shrink, and it likely was shrinking from all of this other therapy rather than shrinking from what the drug had done. And the benefit of this decrease is attributed to the drug as opposed to being attributed to this. And this was actually a true case where the patient had evidence of this effect, and it was the only patient that got a benefit in the study. And they expanded the study just because of this patient, and they didn't see any other replication of it. And it was a waste of money and a waste of patients being involved in a setting like this. So I wanted to get about this idea how a single-arm study can lead to an overrepresentation or an underestimation of the effect of a therapy. So this is a study that was using two drugs, and it was a randomized study. And in this setting, the median survival in recurrent glioblastoma was 9.8 months, and this one was 10 months. So one would say, well, that's our baseline. Our baseline, our historical baseline should be 9 or 10 months. And then this therapy could have come along, which the median survival was 7 months. And if that was just being compared to what we see here as a single-arm study, it would have been a negative study. We would have said, it's actually doing worse than what we thought. But because it was a randomized study, and because the patient population was different than the population that occurred here, this ended up actually being a positive study. That there was almost 2-month improvement in survival, that there was this hazard ratio 0.5, and you could see this benefit that's occurring down here with this patient population. So the difference that occurs between here is those that maybe have a worse outcome with a single arm, have larger tumors, older age, more steroids, lower Cloughesy performance, more IDH, several recurrences done at multiple institutions, versus those that usually have a better outcome that are smaller tumors, younger age, less steroids, higher Cloughesy performance status, IDH mutant, limited recurrence, and all at a single institution. These things can all affect single agent or single arm studies and could give us the wrong idea. Again, for me, the biggest problem is we would have missed that this was an effective therapy here had we not done a randomization. So back to then the types of investigations that we have to do to show that the drugs are actually doing something. Because kind of as I said here, we can go through this process to find out that the drug is safe and just plow forward with this very expensive set of evaluations. So we want to know more than is the drug safe. We want to know, is it doing something? And because of the blood-brain barrier and various things like that, we have to dig a little bit deeper and we have to find out a little bit more about what to expect from this drug before moving it into these later stages. So we go through this process of the phase 1 dose escalation where we give very low doses initially, then we continue, and then we find that this is fine, but we go to this higher group and find there's a problem, patients are having problems, this is the dose we should move forward, and now we go forward and include patients either looking for some kind of signal of response or prolongation of survival, but these are single-arm evaluations, not very helpful, or we simply go from here and go straight into a randomized study. What we do instead, and many of our settings in glioblastoma, is we then go through a process where we identify that there's recurrent tumor. We think this patient's location of the tumor is in a place where we could surgically remove it safely and it would be beneficial. And we give them the treatment, the full dose, the maximum tolerated dose, and then we perform the surgery, and after the surgery is done, they continue on treatment, and we follow them. But this is really the critical area where we're trying to get information. And we're trying to learn, did the drug get in there? Because we have the tumor now, so we can measure drug concentrations, not only in the blood, but in the tumor. Did it hit the target that we're interested in? If it's a EGFR inhibitor, did it inhibit EGFR? If it is an immune therapy, did we see a change in the immune function in the tumor? Sometimes, even in this setting, we have to randomize where people don't get this initial treatment. They just have the recurrent tumor and surgery, but then afterwards, everybody gets treatment. So you can compare the tissue in a setting where they didn't receive treatment or they did receive treatment and try to understand what the benefit is. Because if we see that that happens, if we see that there's a tumor effect, it gives us more confidence moving forward with the phase 2-3 studies that, again, are extremely expensive. Well, there's other ways that we could do it other than doing surgery. You know, one way is that with one of the drugs we're evaluating, if it is effective, it's going to decrease glucose utilization in the tumor. So if we perform a PET scan before, we don't have to do surgery, and we see that there's an uptake, and then we give a specific inhibitor, and in those where we see a decreased utilization of glucose, that's the group that will likely respond or allow us to move forward in a particular way. So by using these approaches, we're much more nimble in the way that we could evaluate the drug, because let's say we used a particular drug here and all of them showed no change in glucose utilization. Well, that would tell us we either have to use higher doses, we have to schedule it so we give 2 days on, 5 days off, do a number of evaluations until we get to the point where we could move forward. Because if we didn't do this, we would just move forward with that phase 2-3 study and find out that we failed because we weren't using the proper dose. So having these early determinants of effect, especially if they're non-invasive, can be really, really helpful for us in developing the drug, understanding we have the right dose and the right schedule, and going forward with phase 2-3 studies. I'll just give you one other example, because sometimes it's hard to tell what's going on with tumors. Are they shrinking? Did we change the rate that they were growing? And we did this in this one study that involved tumors that had lower grade tumors that were IDH mutated, and they got an IDH inhibitor. But before going on the study, we had to show that they had tumor growth coming in. So we go from this historical one to the screening one, and you can see in these areas that the tumor is growing because this empty space is filling in, or it's just simply getting larger. And we could measure the growth rate before going in, and then we start therapy, And then we could see the change in the growth rate. In this case, it's a negative growth rate, but even if it flattened out, we would be convinced that the drug was doing something. So we could see the change after screening, where we see the drug decrease, or the tumor decreasing over time, showing a benefit of the drug. And again, this could be done fairly early on without having to wait a number of years in this case to get that answer, so that we have confidence going forward and committing the funds that are necessary to do a larger study. So that's kind of priming everything. So what we went through to get to the point where we said the drug is doing something, and now we want to show, is it of value to a patient, or can it improve over the standard of care? And ideally, these, normally these are separated as two things, that there's a phase 2 study or a phase 3 study. But what would be ideal is to combine what you would learn not only in the phase 2, but if it was performing well in the phase 2, that it would move to the phase 3, and it would be done all in the same study session. That would save time, because whenever you do these studies, you have to start it up, get all the sites interested, then you enroll them, then you've met your enrollment, you stop enrolling. Now you let these patients' data mature, and then you check and you see how you did. So now there's nobody enrolling, all the sites have stopped, and you said, oh, it looks good. All right, now we have to do this again. Bring all the sites up again, start enrolling, and hit your target of enrollment and shut down again. It's very inefficient. There's lots of what they call white space in between these trials, and that creates inefficiencies. So how can we do something that would allow this all to happen at the same time and be as efficient and effective as possible? So I kind of went through this. I'm going to move to this idea of master protocols and how these can help. And basically, a master protocol is a way of using infrastructure to run trials and have different arms come into the trial and have different arms leave the trial. And to do this, you have to have a common infrastructure. And that means you have to have a common screening setting. The governance of that trial has to be set, where you have steering committees, adjudication committees, monitoring committees. The trial networks and centers have to be in place. And in a sense, what happens is once you have them in place and you have these trials going where you have different arms that could come in and out, they stay active the entire time. You can have common infrastructure with randomization, with data safety, monitoring, quality oversight. All of those can kind of play a role. Then on the design side, there's the opportunity for this adaptive randomization, which I'll get into for you. But if you're collecting data over time on patients, and if you have a platform where arms are coming in and out, you're getting a lot of data, and you're getting this longitudinal data that will help better understand and inform not only how well various arms are doing, but help us better understand the natural history and begin to allow us to kind of look at different ways of evaluating patients, such as growth rates or other things that may be interesting that would give us insight earlier. So there's these areas of infrastructure and trial design, and these are different types of studies that can be done. There's something called umbrella studies. And this has taken a single disease, and this has been used in lung cancer. And with lung cancer, and you could even imagine in breast cancer too, it's separated out into different components of breast cancer. You have HER2 positive. You have estrogen receptor and progesterone receptor positive. You have triple negative. And you can evaluate those different subgroups with different therapies that are appropriate for them and evaluate them in a randomized setting and moving forward. These are called umbrella studies. There are other approaches called basket studies, and that usually is a setting where you have different diseases. You have lung cancer, you have breast cancer, you have brain cancer. They all have the same feature. Those features, then, there's a specific target or drug that goes against those features. And then you could try them all at the same time. Again, this could be controlled or uncontrolled. If it's uncontrolled, it's going to be about tumor shrinkage that you see. If it's controlled, it could be overall survival or other things that would be evaluated. So the final one is this, single disease platform trial. And that's what was developed with GB Imaginal. I kind of want to go through this with you so you have an understanding. So we call it an adaptive platform trial for later stage development in glioblastoma. As I alluded to before, this is a perpetual multi-arm comparative trial. That means there's some control that exists in there. That it goes through 2 phases. And that's why I was kind of saying with phase 2 and phase 3. Phase 2, you try to find out, am I going to learn that there is some benefit to this drug? And if I identify that there's a benefit, I want to confirm it. And so this can happen all in a seamless setting, from going from the first stage into the second stage, and nobody has an idea that this occurs. It uses Bayesian statistics that allow us to determine when we've reached a certain threshold of benefit, and then we could shut down stage 1, go to stage 2, enroll the appropriate number of patients, and this can be used for FDA approval or a new drug application. One of the really cool things about the process of when it reaches a specific threshold is that it's this kind of adaptive approach. And that means you can start off in the beginning with arms all being equally randomized, but as the data mature, it assigns future patients on the best performing arms. That means that if you're a patient going in and you have go into a trial where there's multiple arms and it's been going on, you know that based upon the specific makeup of your tumor and your features, that it would be selecting for you to go into the greatest likelihood, or it would be the greatest likelihood that you would be randomized into the group that is performing the best for your type of tumor, which is really cool, especially because that's something that's never been really done before, certainly in glioblastoma. And then it looks at all indications of glioblastoma, that's first line and recurrence. The idea is that it's international, so that if you get approval and show benefit for FDA approval, that would get approved in China, get approved in Europe, get approved in Australia. And the primary endpoint is overall survival. So here's the value propositions of this. It's the opportunity to test an agent or combinations in an efficient and expedient manner. So the master protocol structure shortens the time to arm initiation because all the sites are ready. So when you bring a new arm in, you're ready to go. So first patient in, it is collapsed. It's cost effective because it is allowing for a rapid, there's a couple different things. The number of patients that are involved makes it cost effective, and the time that it takes from understanding that there's value or no value, you get to learn very quickly. So if you fail early, and that's okay because that tells you that we shouldn't be exploring it anymore, but if you win, you're able to move into the market with the FDA. I'll get into this issue about borrowing power across subtypes, because essentially, if it looks like it's doing well in recurrent and newly diagnosed, those data can be compiled together to show that it's going to move forward. The adaptive randomization identifies the best responsive subtypes, and I'll give you an idea of what that is. But basically, we'll show you how that works. And then there's the possibility of having a companion diagnostic. I'm going to skip by this and just tell you that our primary objectives is that it's to identify experimental therapies that improve overall survival, also to identify specific populations that benefit from these screen therapies, and have those populations confirmed that they're efficacious through a second stage that allows for FDA approval. So here's the way that this therapy and this particular trial would work. We're going to use some of these different terminologies so that I could take you through this. A subtype is a category of disease defined by patient or patient characteristics. A signature could be a population or a collection of subtypes that show that provide a possible indication for therapies. I'll get into this enrichment biomarker. We talked about stage 1 and stage 2. and graduation and fertility, and the graduation simply means it moves on to the next stage. So we're evaluating patients in newly diagnosed methylated, newly diagnosed unmethylated, and recurrent. So there's 3 unique populations. So if a patient comes in, they fit into one of these subtypes. If they have an enrichment marker, let's say the EGFR, in each of these different settings, they could either be EGFR positive or negative in methylated or unmethylated or newly diagnosed or recurrent. So subtypes will take a patient and put them in and say, you fit into this group or you fit into this group. You can't fit into more than one group here because there will be only one group that you would fit into. And if there were multiple experimental arms, some of them having a biomarker, some of them not having a biomarker, and the patient comes in, they're going to be placed and identified as having a particular subtype. So for this experimental arm, they're newly diagnosed methylated with biomarker positive. Here for arm B, it's a different biomarker, they're biomarker negative. Here, they're just newly diagnosed methylated. The same thing across here. So these are the subtypes. A unique patient has a specific subtype. And these subtypes can be put together and create a signature. So if we found out that as we develop data over time on all the different types of patients with different subtypes, that all the patients benefited, then we would be identifying a group of patients and saying that everybody's getting a benefit. So these signatures are evaluated, and essentially if we don't have any biomarkers, we'll just be simply evaluating, is it present in all groups, just newly diagnosed, just unmethylated, just methylated, or in just recurrent. And if we have a biomarker, we add all of these other groups. And the study and the algorithms look to determine, as it's going on, where the drug is performing the best and in what group it's performing the best. So this is what happens. A patient enrolls, we know what the subtype is, they get randomized to the experimental arm or the control, the data are updated, the patients then, we calculate the probability of how the arm is doing, so how the therapy is doing across all the different signatures. And if it's performing well, or if it's doing poorly, we stop for futility. If we reach a maximum number of patients, then we stop and we go no further, or we continue on to get more data, or it graduates and enters stage 2. Entering stage 2 then allows you to randomize to a select group of patients. So we then go ahead and we would add more arms as we need to, but as we update the probability for each of these subtypes and signatures, a new patient will come in. And that new patient will need to be randomized. And now with all this new data, if we find that there's a subgroup that's really doing well for one therapy or another, that new patient then has a higher likelihood of going into the better performing arm. So to some degree, what it looks like is if there were 4 experimental arms and 1 control arm, there's really only going to be 20% of the patients that get on a control arm and 80% that go on the experimental arm. So we take care of the issue that we have around needing a control arm to understand how patients are doing, and yet it's a small percentage of the patients that go on the control arm. And then, of course, the rest are on experimental arms, and we adaptively randomize to the arm that is performing best based upon the subtype of the patient that's coming in next. So one big picture look of how this changes things is there's a couple different ways that we could think about what we want to do. We can say, all right, this is how we're doing this today. We have a control population, we have drug A, there's 3 different populations we want to look at. We're going to use the old style statistics. It's going to be done in multiple countries. It's going to take 1,500 patients. Half of those patients are control. It's going to cost $250 million. Or we could say, we'll do all of these at the same time. At least it's happening faster. We're just going to push this through faster. We have the same number of controls, the same number of drugs. That's now we're looking at 7,500 patients, 5 trials. $1.25 billion. Or we use the same control, we use an adaptively randomized approach that requires less patients. Now we can evaluate 5 arms. We have a common control that each of these drugs borrow against. We use the Bayesian design. We're now down to 1,200 total patients that we need, and the total cost of that goes down to around $125 million. So it's faster, less patients, lower development costs, and we hope that what would come from that, because there's less cost in development, that it would lead to lower consumer costs. You could also imagine that this provides an opportunity for maybe even generics to be evaluated. If someone is willing to pay for those to go through the process, that generics could be evaluated at a much lower level where you could do phase 3 studies and repurpose drugs that have been used for other types of diseases such as hypertension or other approaches like that. So GBM Agile, it didn't just occur. This was a 3 to 4 year process. It involved all these people that are here. This was organized through Webb Cavney, Al Young, and Ann Barker. And Ann Barker played a huge role in putting this all together. But now it is being sponsored through a 501c3 for the operational aspects, where we're involving all different groups to move forward. And we're moving forward with that build now. And we hope to be able to start next year. So I'm going to stop here and see if you have any questions.
Thank you, Dr. Cloughesy. We will now take questions. If you have a question you would like to ask, please type and submit it using the question box in the webinar control panel on the right-hand side of your screen. Our first question for today, Dr. Cloughesy, is for trials like GBM Agile, if a drug doesn't work for a patient, can the patient go on to another drug in the trial, or are they completely out of the trial at that point?
It's an excellent question. So one of the issues, because the overall endpoint is overall survival. So if another therapy that a patient takes is then beneficial, both therapies potentially get credited. That becomes a little bit of a problem, but in the beginning, we're not going to allow patients to be re-randomized. But in the future, what really happens is those patients can then just go somewhere else and go on a trial. So why not keep them in the setting, in the same setting? And I think we just have to work with the FDA to figure out the best way to do that so that they could be confident that when they see an effect from a drug, they can attribute it to the appropriate drug.
Thank you. Our next question, with all the issues that you discussed with single-arm studies, why do they not just always do randomized trials? Why do a single-arm study at all for phase 2?
It's a really good question, but there's two things. One is a lot of these studies are going to be cheaper when you do a single arm. They're going to be sometimes just done at one institution. But the data aren't very good. So the different things that are pushing in that direction are costs and interest in kind of developing it themselves in their local environment. The other issue that comes up is it's hard for patients to be a control. Nobody wants to do it, and yet at the same time, if that's the only way we learn, maybe we could minimize the impact of being a control like we have in GBM Agile. So instead of having 3,500 controls to evaluate 5 different drugs, we end up with 200 to 250 controls that are needed.
Thank you. Our next question, is the Agile method being used on any other tumor types, and are they being used on any pediatric tumors?
So there's approaches that are going forward. So this approach, at first, the screening part was used in I-SPY 2, developed by the same statisticians, the Berry consultant group. But it wasn't, I-SPY 2 was not a registration study. It couldn't go and function like a phase 3. So this is different in that it functions like a phase 3. But there are other approaches moving forward, like with pancreatic cancer, where that is occurring. So it's an approach that could be used with other tumor types, especially those tumor types that aren't getting the obvious benefit from the current clinical trial approach.
And what current drugs are being considered for Agile, and how are they selected?
So the drugs really, as maybe that earlier slide showed, we look at any drug. We're agnostic to any drug. We do have a selection committee that is involved in understanding whether we should be considering this. And the types of things that they look at is, how specific is the drug for glioblastoma? What data exist that are in models? Do we have any data that are in the clinic yet with patients that can be evaluated? All of these things are taken into account. And the success of this approach, let's say, in other tumor types is also taken into account. Like immune therapies have been successful in melanoma, not so much in brain cancer. But if it hadn't been used in brain cancer, we would want to evaluate that because we'd want to take something that's successful and make sure whether or not it works in brain cancer.
Our next question is, what is the criteria, or what needs to happen for a clinical trial to move from the animal experiment phase to human groups?
Yeah, so there's a couple of different things. I think part of it, there is a lot of money that has to go into getting the drug ready to be clinically used. And that means that you gotta then have a lot of confidence about the evaluations that you do preclinically, either in animals or in cell lines. And the more the cell lines are relevant to the disease, the better. We have some very old cell lines that we've used, in the past, like U87. These were probably developed in 1950. They continue on as the lines could be passaged, but they've changed so much that they have nothing to do with brain cancer anymore. So using lines and models that have the greatest likelihood of mimicking human brain cancer is ideal. And there just has to be, and there isn't any particular threshold, but because the financial investment is significant to make clinical quality drug and to do all the tests that have to be done in animals before it's allowed to go into humans, the data need to be pretty compelling.
Do you have any suggestions for how someone can get involved in a clinical trial, or how they can find one that's either in their community or that they would qualify for?
Yeah, so there's a lot of different clinical trials that exist, and the hard thing is, the phase 1 trials open and close. The phase 2 trials and 3 trials should be open for longer periods of time. The clinicaltrials.gov, I think, is a great way to go about and get that kind of information. I don't know, you may want to share if you have groups through ABTA that you use to help people find trials. But we hope that what will also happen with GBM Agile is that there'll be enough sites that will be open throughout the US and throughout the world that will allow patients to have more access. And because it will always be open, it's just a matter of just going, and that the algorithms that are set up will do the best job it can to select the best therapies for a patient.
Thank you for sharing that. And just to kind of follow up on your response, the ABTA does have a program called Trial Connect, which you can find on our website, which helps match patients with clinical trials in their communities that they would qualify for. I do think that is all the time that we have for today. Thank you all for joining us, and thanks once again to Dr. Cloughesy for his wonderful webinar presentation. Besides our free educational webinars, the ABTA has a variety of programs available to connect patients and caregivers with information and resources to help support them in their brain tumor journey, as well as publications and resources for healthcare professionals. For more information, visit ABTA's website at www.abta.org or call the ABTA Care Line at 1-800-886-2282. We will now end our webinar.